Chapter 15, Plant Growth and Development. You have already studied the organization of a flowering plant in Chapter 5. Have you ever thought about where and how the structures like root systems, leaves, flowers, fruits, and seeds arise, that too in a orderly sequence? You are by now aware of the terms seeds, seedling, plantlet, mature plant. You have also seen that trees continue to increase in height or girth over a period of time. However, the leaves, flowers, and fruits of the same tree not only have limited dimension but also appear and fall periodically and some time repeatedly. Why does vegetative phase precede flowering in plant? On plant, all plant organs are made up of a variety of tissues. Is there any relationship between the structures of a cell, a tissue, an organ, and the function they perform? Can the structure and the function of these be altered? All cells of a plant are descendants of the zygote. The question is then why and how do they have a different structural and functional attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits, and seeds, and eventually they die. Figure 15.1. The first step in the process of a plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of such favorable conditions, the seed do not germinate and goes into a period of suspended growth or rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. In this chapter, you shall also study some of the factors which govern control these developmental processes. These factors are both intrinsic, internal, and extrinsic, external to the plant. Figure 15.1, Germination of seedling development in bean. Soil line, cotyledon, hypocotyle, seed coat, epicotyle hook, cotyledons, hypocotyle, hypocotyle. 15.1, Growth. Growth is regarded as one of the most fundamental and conspicuous characteristic of a living being. What is growth? Growth can be defined as an irreversible, permanent increase in size of an organ or its part, or even of an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, an example, expansion of a leaf of a growth. How would you describe the swelling of a piece of a wood when placed in water? 15.1.1, Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of the plants is due to the presence of meristems at certain locations in their body. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. This form of growth, where new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? In chapter 6, you have studied about the root apical meristem and the shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contribute to the elongation of the plant along their axis. You also know that in dicotyledonous plants and gymnosperms, the lateral meristems, vascular cambium, and corpus cambium appear later in life. These are meristems then causes the increase in the girth of the organs in which they are active. This is known as secondary growth. Figure 15.2, diagrammatic representation of the location of shoot apical meristem. Shoot apical meristem and vascular cambium allows exhibit arrows exhibit the direction of growth of cells and organs. Shoot apical meristem, vascular cambium, root apical meristem. 15.1.2, Growth is measurable. Growth at all cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in the watermelon may increase in size by up to 350,000 times. In the former, growth is expressed as increase in cell number. The latter expresses the growth as increase in the size of the cell, while the growth of the pollen tuberose is measured and is measured in terms of its length. An increase in surface area denotes the growth in a dorsiventral leaf. 15.1.3 phases of growth. A period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. Figure 15.3 detection of zones of elongation by the parallel line technique. Zones A, B, C, D immediately behind the apex have elongated most. A, B, C, D, E, F, G. Let us understand this by looking at the root tips. The constantly dividing cells, but both at the root apex and the shoot apex represent the meristematic phase of growth. The cells in this region are rich in protoplasm, possess large conspicuous nuclei. The cell walls are primarily in nature thin and cellulosic with abundant plasmodesmatal connections. The cells proximal just next away from the tip to the meristematic zone represent the phase of elongation. Increased vacuolation, cell enlargement, and new cell wall deposition are the characteristic of the cells in this phase. Further away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of the zone attaining their maximal size in terms of wall thickening and protoplasmic notifications. Most of the tissues and cell types you have studied in chapter 6 represent this phase. 15.1 Growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically as an organism or a part of the organism can produce more cells in a variety of ways. 15.4 Figure diagrammatic representation of A. Arithmetic, B. Geometric growth, and C. Stages during embryo development showing geometric and arithmetic phases. Zygote divided, geometric phase, all cells divided. Arithmetic phase, cells capable of division, cells that lose capacity to divide. The growth ratios are increased that may be arithmetic or geometrical. In arithmetic growth, following mitotic cell division, only one daughter cell continues to divide, while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongating at a constant rate. Look at figure 15.5 on plotting the length of the organ against time. A linear curve is obtained mathematically. It is expressed as LT is equal to L0 plus RT. LT is length at time t, L0 is length at time 0. R is growth rate or elongation per unit time. 15.5 figure constant linear growth, a plot of length L and against time t. Height of the plant, time. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow, lag phase, and it increases rapidly thereafter at an exponential growth, log or exponential phase. Here, both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S curve. A sigmoid curve is a characteristic of living organism growing in a natural environment. It is typical for all cells, tissues, and organs of a plant. Can you think of more similar examples? What kind of a curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed as W1 is equal to W0 E R raised to RT. W1 is final size, weight, height, number, etc. W0 is initial size at the beginning of the period. R is the growth rate, T is the time of growth, E is the base of natural logarithms. R is the relative growth rate and is also the measure of the ability of the plant to produce a new plant material, referred to as efficiency index. Hence, the final size of W1 depends on the initial size W0. 15.6 figure. An idealized sigmoid growth curve typical of cells in culture and many higher plants and plant organs. Size or weight of the organ, time, lag phase, exponential phase, and the stationary phase. Quantitative comparison between the growth of living systems can also be made in two ways. First, measurement and the comparison of total growth per unit time is also for absolute growth. Second, the growth of the given system per unit time expressed on a common basis. For example, per unit, a initial parameter is called the relative growth rate. In figure 15.72, leaves A and B are drawn that are of different sizes but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them shows a much higher relative growth rate, which one and why. Figure 15.7 diagrammatic comparison of absolute and relative growth rates. Both leaves A and B have increased their area by 5 cm in a given time to produce A1, B1 leaves. Leaf A, leaf B. 15.1.5 Conditions for growth. Why do you not try to write down what you think are necessary conditions for growth? This list may have water, oxygen, and nutrients as very essential elements for growth. The plant cells grow in size by cell enlargement, which in turn requires water. Rigidity of cells helps in extensive growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients, macro and micro essential elements are required by plants for the synthesis of protoplasm and acts as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range would be detrimental to its survival. Environmental signals such as light and gravity also affect certain phases or stages of growth. 15.2 Differentiation, de-differentiation and redifferentiation. The cells derived from root apical and shoot apical meristem and cambium differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell walls and protoplasm. For example, to form a trachyary element, the cells would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distances. Even lignocellulosic secondary cell walls carry water to long distances. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distances even under extreme tensions. Try to correlate the various anatomical features you encounter in plants to the functions they perform. Plants show another interesting phenomenon. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomenon is termed as de-differentiation. For example, formation of meristems interfascicular cambium and cork cambium from fully differentiated parenchyma cells. While doing so, such meristems or tissues are able to divide and produce cells that once again lose the capacity to divide but mature to form specific functions, that is, get re-differentiated. List some of the tissues in a woody dicotyledonous plant that are the products of re-differentiation. How would you describe a tumor? What would you call the parenchyma cells that are made by divided under controlled laboratory conditions during plant tissue culture? Recall in section 15.1.1, we have mentioned that the growth in plants is open, that is, it can be indeterminate or determinate. Now we may say that the same meristem have different structures at maturity. The final structure and maturity of a cellular tissue is also determined by the location of the cell within. For example, cells position away from the root apical meristem differentiate as a root cap cells, while those pushed to the periphery mature as epidermis. Can you add a few more examples of open differentiation correlation? The position of a cell to its position in an organ. 15.3 Development. Development is a term that includes all changes that an organism goes through during its life cycle, from germination of the seeds to senescence. Diagrammatic representation of the sequence of processes which constitute the development of a cell of a higher plant is given in figure 15.8. It is also applicable to tissues or organs. 15.8 Sequence of the developmental process in a plant cell. Sequence of the developmental process in a plant cell: meristematic cell, cell division, plasmatic growth, differentiation, expansion, elongation, maturation, mature cell, senescence. Plants follow different pathways in response to environments or phases of life to form different kinds of structures. This ability is called plasticity. For example, heterophyll in cotton, coriander, larkspur. In such plants, the leaves of the juvenile plant are different in shape from those in mature plant. On the other hand, difference in shape or leaves produced in air and those produced in water in buttercup also represent the heterophylous development due to the environment. This phenomenon of heterophyly is an example of plasticity. 15.9 Figure heterophyly in larkspur, juvenile phase, adult phase, B, buttercup, terrestrial habitat, water habitat. Thus, growth, differentiation, and development are very closely related events in the life of a plant. Broadly, developmental is considered as the sum growth of differentiation development in plants, that is, both growth and differentiation is under the control of intrinsic extrinsic factors. The former includes both intracellular, genetic, or intercellular factors, chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrition, etc. 15.4 Plant growth regulators 15.4.1 characteristics. The plant growth regulators PGRs are small simple molecules of diverse chemical composition. They could be compounds indole. 3 acetic acid IAA, adenine derivatives, and 6 furfuryl amino purine, kinodine derivatives of carotenoids, abscisic acid, ABA, terpenes, gibberellic acid, GE3 or gases, ethylene, C2H4. Plant growth regulators are variously described as plant growth substances, plant hormones, or phytohormones in literature. The PGRs can be broadly divided into two groups based on their functions in the living plant body. One group of the PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are called as the plant growth promoters, for example, auxins, gibberellins, and cytokinins. The PGRs of the other group play an important role in plant response to wounds, stress, and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. 15.4.2 The discovery of plant growth regulators. Interestingly, the discovery of each of the five major groups of the PGRs have been accidental. Also, they started with the observation of Charles Darwin and his son Francis Darwin when they observed that the coleoptiles or canary grass responded to unilateral illumination by growing towards the light source. Phototropism. After a series of experiments, it was concluded that the tip of the coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Auxin was isolated by F.W. Wendt from tips of coleoptiles of oat seedlings. Figure 15.10 experiments used to demonstrate the tip of the coleoptile is the source of auxin. Arrows indicate direction of light. A, B, C, D. The Bacchanae foolish seedling disease of rice seedlings was caused by a fungal pathogen Gibberella fujikuraya. E. Kurosawa 1926 reported the appearance of the symptoms of the disease in rice seedlings when they were treated with the sterile filtrates of the fungus. The active substances were later identified as gibberellic acid. F. Skoog and his co-workers observed that from the internodal segments of tobacco stems in the callus, a mass of undifferentiated cells proliferated only if, in addition to auxins, the nutrient medium was supplemented with one of the following extracts of the vascular tissue: yeast extract, coconut milk, or DNA. Skoog and Miller later identified and crystallized the kinases promoting active substances that they termed kinetin. During mid-1960s, three independent researchers reported the purification and chemical characterization of three different kinds of inhibitors: inhibitor B, abscission 2, abscission 2nd, and dormin. Later all the three were proved to be chemically identical. It was named abscisic acid ABA. Cousins confirmed the release of a volatile substance from a ripened orange that hastened the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effects of these five categories of the PGRs in the next section. 15.4.3 Physiological effects of plant growth regulators. 15.4.3.1 Auxins. Auxins, from Greek auxine, to grow, was first isolated from human urine. The term auxin is applied to the indole 3-acetic acid IAA and to other natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stems and roots, from where they migrate to the regions of their actions. Auxins like IAA and indole butyric acid IBA have also been isolated from plants. NAA, naphthalene acetic acid, and 2,4-D, that is 2,4-dichlorophenoxyacetate, are synthetic auxins. All these auxins have been used extensively in the agricultural and horticultural practices. They help to initiate rooting and stem cuttings, and application are widely used for plant propagation. Auxins promote flowering, for example, in pineapples. They help to prevent fruit and leaf drop at early stages, but promote the abscission of older mature leaves and fruits. In most higher plants, like the growing apical bud inhibits the growth of the lateral axillary buds, a phenomenon called apical dominance. Removal of shoot tips or decapitation usually results in the growth of lateral buds. It is widely applied in tea plantations. Hedge making can explain why. Auxins also induce parthenocarpy, for example, in tomatoes. They are widely used as herbicides. 2,4-D, widely used to kill dicotyledonous weeds. Does not affect mature monocotyledonous plants. It is used to prepare weed-free lawns by gardeners. Auxin also controls xylem differentiation and help in cell division. Figure 15.11, apical dominance in plants. A. Plant with apical bud intact. B. A plant with apical bud removed. Note the growth of lateral buds into branches after decapitation. 15.4.3.2, gibberellins. Gibberellins are another kind of promotory PGR. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2, and GA3, and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remains the most intensively studied form. All GAs are acidic. They produce a wide range of physiological responses in the plants. Their ability to cause an increase in length of axis is used to increase the length of grapes stalks. Gibberellins cause fruits like apples to elongate and improve its shape. They also delay senescence, thus the growth can be left on the tree longer so as to extend the market period. GA3 is used to speed up the malting process in brewing industry. Sugarcane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellins increases the length of the stem, thus increasing the yield by as much as 20 tons per acre. Spraying juvenile conifers with GAs hastens the maturity period, thus leading to early seed production. Gibberellins also promotes bolting and internode elongation just prior to flowering. In beet, cabbages, and many plants with rosette habit. 15.4.3.3, cytokinins. Cytokinins have specific effects on cytokinases and were discovered as a kinatin, a modified form of adenine purine from the autoclaved herring sperm DNA. Kinatin does not occur naturally in plants. Search for natural substances with cytokinin-like activities led to the isolation of zeatin from corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in the regions where rapid cell division occurs. For example, root of ice is developing shoot buds, young fruits, etc. It also helps to produce new leaves, chloroplast in leaf, lateral shoot growth, and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote a neutron mobilization, which helps in the delay of leaf senescence. 15.4.3.4, ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amounts by tissues undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plant organs, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. This rise in the rate of respiration is called respiratory climactic. Ethylene breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubers. Ethylene promotes the rapid internode or petiole elongation in deep water rice plants. It helps the leaves or upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also includes flowering in mango. Since ethylene regulates so many physiological processes, that is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethaphone. Ethaphone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Ethaphone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnut. It promotes female flowers in cucumbers, thereby increasing the yield. 15.4.3.5, abscisic acid. As I mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy, but like other PGRs, it also has other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of a plant metabolism. ABA inhibits its seed germination. ABA stimulates the closure of the stomata and the epidermis and increases the tolerance of plant to various kinds of stresses. Therefore, it is also called the stress hormone. ABA plays an important role in seed development, maturation, and dormancy. By inducing dormancy, ABA also helps the seeds to withstand desiccation and other factors unfavorable for growth. In most situations, ABA acts as an antagonist to GAs. We may summarize that for any and every phase of the growth, differentiation, and development of plants. One or the other PGR has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect the event. For example, dormancy in seeds or buds, abscission and senescence, apical dominance, etc. Remember, the role of the PGR is of only one kind of intrinsic control. Along with genome control, the intrinsic factors they play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via PGR. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly the role of light and temperature, both of them extrinsic factors on initiation of flowering. 15.5 Photoperiodism. It has been observed that some plants require a periodic exposure to light to induce flowering. It is also seen that such plants are able to measure the duration of exposure to light. For example, some plants require the exposure to light for a period exceeding a well-defined critical duration, while others must be exposed to light for a period less than this critical duration before the flowering is initiated in them. The former group of plants are called long day plants, while the latter ones are termed as short day plants. The critical duration is different for different plants. There are many plants, however, where there is no such correlation between exposure to light duration and induction of flowering response. Such plants are called day-neutral plants. Figure 15.12. Photoperiodism, long day, short day, and day-neutral plants. Long day plant, flowering, but critical photoperiod, below, no flowering. Short day plant, flowering, below, critical photoperiod, no flowering, above. Day-neutral plant, flowering after vegetative maturity. It is now also known that not only the duration of the light period but that of the duration of dark period is also of equal importance. Hence, it can be said that flowering in certain plants depends not only on a combination of light and dark periods of day or night is termed as photoperiodism. It is also interesting to note that while shoot apices modify themselves into flowering apices prior to flowering, that is shoot apices of plants by themselves, do not perceive photoperiods. The site of perception of light or dark duration or the leaves, it has been hypothesized that there is hormonal substances that is responsible for flowering. This hormonal substance migrates from leaves to shoot apices for inducing flowering only when the plant are exposed to the necessary inductive photoperiod. 15.6 Vernalization. There are plants for which flowering is either quantitative or qualitative dependent on exposure to low temperature. This phenomenon is termed as vernalization. It prevents precocious reproductive development late in the growing season and enables the plant to have a sufficient time to reach maturity. Vernalization refers especially to the promotion of flowering by a period of low temperature. Some important food plants, wheat, barley, rye, have two kinds of varieties, winter and spring varieties. The spring variety are normally planted in the spring and come to the flower and produce grain before the end of the growing season. Winter varieties, however, if planted in spring, would normally fail to flower or produce mature grain within a span of a flowering season. Hence, they are planted in autumn, they germinate and over winter come out as a small seedlings, resume growth in the spring, and are harvested usually around the midsummer. Another example of vernalization is seen in biennial plants. Biennials are monocarpic plants that are normally flower and die in the second season. Sugar beet, cabbages, carrots are some of the common biennials. Subjecting the growing of a biennial plant to a cold treatment stimulates a subsequent photoperiodic flowering response. 15.7 Seed dormancy. There are certain seeds which fail to germinate even when the external conditions are favorable. Such seeds are understood to be undergoing a period of dormancy which is controlled not only by external environment but are under endogenous control or conditions within the seed itself. Impermeable and hard seed coat, presence of chemical inhibitors such as abscisic acid, phenolic acids, parascorbic acid, and immature embryos are some of the reasons which cause a seed dormancy. This dormancy, however, can be overcome through natural means and various man-made measures. For example, the seed coat barrier in some of the seeds can be broken by mechanical aberrations using knives, sandpaper, etc., or vigorous shaking in nature. These aberrations are caused by microbial action and passage through digestive tract of mills. Effective inhibitory substances can be removed by subjecting the seeds to chilling conditions or by application of certain chemicals like gibberellic acid and nitrates. Changing the environmental conditions such as light and temperature Other methods to overcome seed dormancy. Summary: growth is one of the most conspicuous events in a living organism. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involves increased protoplasmic material in plants meristums at the site of growth, root and shoot. Apical meristem, sometimes along with intercalary meristem, contribute to the elongation growth of the plant axis. Growth is indeterminate in higher plants. Following cell division in root and shoot apical meristem cells, the growth could be arithmetic or geometrical. Growth may not be and generally is not sustained at a higher rate throughout the life of a cell or a tissue or organ or organism. One can define three principal phases of growth: the lag, the log, and the senescent phase. When a cell loses the capacity to divide, it leads to the differentiation. Differentiation results in the development of structures that is commensurate with the function the cell finally has to perform. General principles for differentiation for cell, tissues, and organs are similar. A differentiated cell may be de-differentiate and then pre-differentiate. Since differentiation in plants is open, the development could also be flexible. That is, the development is the sum of growth and differentiation. Plants exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factors. Intercellular intrinsic factors are the chemical substances called plant growth regulators, PGR. There are diverse groups of PGRs in plants, principally belonging to five groups: auxins, gibberellins, cytokinins, acetic acid, ethylene. These PGRs are synthesized in various parts of the plant. They control different differentiation and developmental events. Any PGR has diverse physiological effects on plants. Diverse PGRs also manifest similar effects. PGRs may act synergistically or antagonistically. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such as external factors. Flowering in some plants is induced only when exposed to certain duration of photoperiod. Depending on the nature of the photoperiod requirements, the plants are called short-day plants, long-day plants, and day-neutral plants. Certain plants also need to be exposed to low temperature so as to hasten flowering later in life. This treatment is known as vernalization.